DNA during the cell cycle. Hi everyone, you've been learning a lot already about how one parent cell divides into two daughter cells. What we haven't talked about, though, is how that parent cell ensures that a copy of its DNA ends up in both daughter cells at once. That's a pretty tricky process, and this video is here to explain how it does so. In eukaryotic cells during interphase, that is most of the cell cycle, G1, S phase, and G2, the DNA is kept inside the nucleus. And you can see a nuclear envelope around the outside of this nucleus right here, even complete with nuclear pores. And the DNA is kept in its active but unwound form called chromatin. Chromatin is a huge scrambled mess like you can see right here. If you were to zoom in on one of these tiny strings, you'd actually be able to read the genetic code inside the double helix of the DNA. But from a more zoomed out perspective, it looks like a messy plate of spaghetti. This is perfect for when the cell is active because this little chunk of DNA can be read just as well as this little chunk of DNA, as well as this chunk of DNA. And so, in this form, the chromatin allows the nucleus to be a very good boss of the cell. This doesn't work so well, though, when the cell is trying to divide, because this is a huge mess. The cell needs to get a lot more organized about it. So during cell division, it takes that chromatin and it organizes it into chromosomes. If you picture taking this long, skinny bit of DNA and wrapping it around some proteins and then wrapping that string around itself and then wrapping those coils around themselves, eventually you can form this large X-like structure known as a chromosome. And chromosomes are much easier to move around than that huge plate of spaghetti. So this is a very desirable shape to keep your DNA in if you're in the middle of dividing. Now, chromosomes come in two forms, an unduplicated form and a duplicated form. Just like it's used in English, to duplicate means to copy. So the DNA on this half of the chromosome is identical in sequence to the DNA that's on this half of the chromosome. The two halves of this duplicated chromosome are called sister chromatids. Though we can't really see it, during S phase of interphase, this unduplicated chromosome is actually being made into a duplicated chromosome as the DNA is copied. And then once cell division begins, we can actually see that it's this duplicated form of the chromosome that condenses out of the chromatin rather than the unduplicated type right here. Let's clarify some of these terms. Picture for a moment we have an unduplicated chromosome. If the cell with this chromosome goes through S phase, where an extra copy of the DNA is made, then we come out with a duplicated chromosome. If you take a duplicated chromosome and you subtract one sister chromatid, remember a sister chromatid is one half of a duplicated chromosome, you get an unduplicated chromosome left over. One final way to look at a duplicated chromosome is by its parts. While this might look like an unduplicated chromosome, since it's actually part of one larger duplicated chromosome and it's just half of it, we call it a sister chromatid. So if you add two sister chromatids together and you attach them using this small central piece called a centromere, that's how you create yourself a duplicated chromosome. So here's some real pictures of chromosomes. Can you tell whether they're duplicated or unduplicated? If I draw in the structures, it might be even more obvious. A centromere in the center, one sister chromatid right here, another sister chromatid right here. They're both attached together, and so this is a duplicated chromosome. So now that we've learned all the different forms that DNA comes in, chromatin, chromosomes, duplicated or unduplicated, chromatids, let's look at which forms of DNA are found at which phases of the cell cycle. So first off, in interphase, which is this whole green slice of the diagram right here, the DNA is kept only in chromatin form, so it's all unwound, very easy to read, but not very organized, nor easy to divide. Once prophase of mitosis has begun, however, the DNA has condensed now into duplicated chromosomes. During metaphase of mitosis, the chromosomes are still duplicated, only now the spindle fibers have lined them up along the middle of the cell. And you can see that one sister chromatid might be straddling in an eastward direction, while the other one is sort of straddling the midline in a westward direction. Then, during anaphase, the transition occurs. Those sister chromatids that were attached into a duplicated chromosome have now broken apart into their own individual unduplicated chromosomes. Those unduplicated chromosomes are dragged to either end of the cell. Those unduplicated chromosomes are then each surrounded by their own nuclear envelope, and the chromosomes can start to unwind into chromatin, and then by the time cytokinesis has occurred, the nuclei are just filled with a big mess of chromatin again that can be used for the rest of interphase, and round we go. The cool thing about chromosomes is that they're found in us. Here are some of my own self-grown organic chromatin and chromosomes. Now, because I'm a total nerd, I have worked in a bunch of labs, and one of the places I was working, I had somebody walk up to me and say, "Hey, we're doing a study, we really need a normal male negative control." And I was like, "I don't know why you think I'm normal, but please, by all means, take my blood, as long as you give me pictures of my cells." And so these people took my cells. Most of the time they look like this with a whole bunch of chromatin inside. It's all messy. But then they ran some of my cells through their cell cycle, and when they got to metaphase, they stopped the cell and they killed them, and they smashed the cell open, and then they took pictures of all the chromosomes inside, and here are my actual chromosomes. Now that last picture was a big mess, so instead they use a bunch of Photoshop and they chop out different parts of the picture and rearrange them in pairs based on which chromosome is which. So you can see all 46 of my chromosomes right here. So the fact that I have 46 chromosomes suggests that I'm actually human, which is sort of strange because all humans have 46 chromosomes. And you can see I got 2 chromosome 1s and 2 chromosome 2s, one from mom, one from dad. And then if you get down to the end here, you'll even find that I have an X chromosome right here and a Y chromosome right here, suggesting that I am in fact a man. Sometimes when you're looking at diagram after diagram of cells and chromosomes, it gets a little tiresome and you wonder why you should care about either of them. However, when I'm looking at a karyotype, these organized diagrams of every single chromosome inside a human being, specifically my own karyotype, I just get really excited. I think, oh my gosh, every one of these chromosomes contained some of the code that makes me, and if a single one of them was missing, I would not be the same. In fact, I could probably not survive. And so thank God that that original parent cell, back when I was 1 cell big, was able to run its cell cycle and mitosis so precisely so as to make extra copies of that DNA and be sure that every single daughter cell of the trillions of cells in my body has 1 copy of every single chromosome.